So let's continue here, and this time here we're going to take a look at the aerobic pathways. And so the aerobic pathways are going to come about because of the fact that we don't stop doing activity after 180 seconds. And we don't always have very high demands for ATP. So what happens when we have to continue to regenerate ATP, and what happens when ATP demands are low? Well, we're going to go about using the more efficient pathway, the aerobic pathway. The aerobic pathway is going to take place for long periods of time. It's going to take place within the mitochondria, and it's going to combine substrate level phosphorylation with oxidative phosphorylation using two linked systems. The ETC, the electron transport chain, coupled with the Krebs cycle or the TCA cycle. We can think of these as being inexhaustible as long as we have fuel sources available, the energy carrying molecules available, oxygen available, and the availability of coenzymes within the ETC. So we're able to go about getting 180 seconds of activity going about. But now we're going to go about and continue to do more work. So we're going to take that pyruvate, and instead of sending it into lactate metabolism, we're going to allow it to become acetyl-CoA. We're going to import some of the other fuel sources available to us. As the NADH plus and the FADH2 are formed from the Krebs cycle, they're going to be shuttled into the electron transport chain, where in combination with transporters and oxygen, they're going to be able to oxidatively phosphorylate ADP into ATP, regenerating NAD and FAD for Krebs and glycolysis to continue. So let's take a look here first at the Krebs cycle. The Krebs cycle is going to take place within the cisternal spaces of the mitochondria, or the open space within the mitochondria. It is required to have oxygen present. However, oxygen is not involved with the system. It's going to cycle one pyruvate, which means that for every glucose that comes in, we get 2 cycles. We're going to be producing 1 ATP, 3 NADH pluses, and 1 FADH2 per turn of the cycle. And so when we look at the cycle, remember we're going to have this duplicated in terms of what gets formed. Now, notice I said that there has to be oxygen available, but it's not actually involved with the system. The reason why we need oxygen available is because we have to be constantly cycling NADs back and FADs back, and without clearing them back via the electron transport chain, the Krebs cycle will stop functioning. The other thing that will stop the Krebs cycle from functioning is a backlog on the fore end of the system. So let's take a look at the Krebs cycle here. And so the Krebs cycle is going to be a cyclic action where we're going to be forming a 6-carbon sugar and then slowly removing carbons, leading to the formation of a 4-carbon sugar, which will then form back to form the 6-carbon sugar. And so let's take a look at each one of these steps along the way. What I've done is I have highlighted the areas where the reaction is taking place with the formation of acetyl-CoA from pyruvate. So each stage of the cycle will be linked somehow with the formation of NADH plus or FADH2 that's going to come out as a byproduct. That NADH plus and FADH2 will be transported in the electron transport chain and recycled back to the Krebs cycle to continue the Krebs cycle within the mitochondria. So the first step here is reaction of acetyl-CoA and OAA, oxaloacetate, with an enzyme known as citrate synthase to form a molecule citrate. Cleaving away the coenzyme A, that coenzyme A will go into what's referred as the CoA pool, which will be used by the mitochondria. The mitochondria needs CoAs, and that need of CoA is going to lead to other issues that we'll talk about with different types of metabolism here. The first reaction is an isomerization reaction in which we take citrate and form it into isocitrate. Isocitrate will then undergo a dehydration reaction. We're going to be removing a carboxyl group, and by removing our carboxyl group, we're going to be removing hydrogens. In this, we're going to form NADHH+, form CO2 as byproducts of the reaction, and form alpha-ketoglutarate as the next step in the cycle. Alpha-ketoglutarate will undergo two. It's going to undergo a dehydrogenasing reaction, followed by an acetylating reaction in which the CoA, which was in the CoA pool, will get junctioned onto the acetyl-CoA, in which we're going to form two distinct byproducts here. We're going to form NADHH+ and CO2 in the first step, and we're going to form GTP. Now, the mitochondria doesn't want to get rid of that GTP, and so what's going to do is it's going to take the GTP and transfer the phosphate onto an ADP to form ATP. We're going to shear away the CoA from the succinyl-CoA that had been formed. In this step here, we're going to go about forming succinate. Succinate undergo a dehydrogenase reaction in which we're going to form FADH2 as the byproduct, forming fumarate as our next step in the pathway. Fumarate will then undergo a hydration reaction via ferramase to form the sugar malate. Malate will then undergo a dehydrogenase reaction, forming NADHH+, recycling oxaloacetate, OAA. Now, within each of the steps of the Krebs cycle, we can indicate them as the sugar, like I've done in the presentation here, or by the acid that is sometimes referenced, hence the TCA cycle. The A in that stands for acid. And so let's take a look at the cycle and indicate where we're going to have rate-limiting steps here. So the first rate-limiting step is the transference of pyruvate into the mitochondria and the formation of acetyl-CoA. The next rate-limiting step is the formation of citric acid. And then the last of the rate-limiting steps is going to be on the fore end of the cycle, forming alpha-ketoglutarate and succinyl-CoA, where increased amounts of malate and furamate or formic acid leads to a backlog and slows down the rate at which the cycle will continue. If the cycle slows down, instead of continuing with Krebs cycle, we will start to undergo lipogenesis, making of lipids. Now, one of the things we have to do is we have to somehow clear the NADHH pluses and the FADH2s so that we can get the NADs and the FAD back so that we can go about continuing the Krebs cycle. We have about 3 to 5 minutes worth of available oxygen to allow for the ETC to continue without having backlog on the Krebs cycle. What the ETC is going to do, with the electron transport chain or the electron transport system is going to do, is it's going to do a process known as oxidative phosphorylation. This is where we are not simply just transferring phosphates, but instead utilizing an enzymatic chain of redox reactions that will eventually allow for an ATP synthase to be stimulated. The way in which this functions is it functions via hydrogen pump action. The hydrogen pump creates what's referred to as an a electrochemical gradient of hydrogens and electrons around the internal membrane of the mitochondria. This develops enough energy within the membrane to power up an ATP synthase, which will allow for the phosphorylation of ADP into ATP. We have a series of stepwise coenzymatic and cytochromatic reactions that are passing the hydrogens and electrons along, in which oxygen will be a terminal reduced agent. It's going to gain the electrons and the hydrogens, forming water. The ETC has two products being formed, ATP and water. This is the only pathway where we can consider ATP to be a product of the reactions. So let's take a look at this stepwise process that's going to couple the stored chemopotential energy with the production of ATP. From the NADH+, we will be able to generate about 3 moles of ATP. From the FADH2, we will be able to generate about 2 moles of ATP. Now, that total mole quantity is going to vary based off of how we discuss the balance of the equation. Most biochemical reactions and most biochemical references give about 2.5 moles of ATP per NADH+, and 1.5 moles of ATP per mole of NADH2. And that has to deal not necessarily with the stoichiometric balance, but with the energetic balance. And so we have 4 distinct pathways or systems that are going to be involved here. And we simply number them 1, 2, 3, and 4. System 1 interacts with NADH+, stripping the hydrogen, which gets passed along. The NAD goes back to where it came from, either Krebs or glycolysis. The hydrogen gets passed along. We interact with system 2. The interaction between system 1 and system 2 activates enough energy to activate the ATP synthase, and we get our first ATP from NADH+. System 2 is the CoQ system. Secondary to system 1, we have a other receptor for the hydrogens, which is going to strip the FADHs of their hydrogen. CoQ will then pass along the hydrogen to complex 3, which passes the hydrogen along. We get enough chemoelectrical charge to power up an ATP synthase. Hydrogen will be passed along to system 4, where once again we will generate enough chemoelectrical potential to power up the ATP synthase. The hydrogens at the terminus here at cytochrome A3 will lock onto oxygen that is being bound to that area, forming water. Now, within this pathway, we have distinct areas where radicalization can cause issues. Because we're passing a radicaled ion along a series of redox reactions, we have to somehow stabilize the membrane and stabilize the products. There are 4 distinct functional units that will act as antioxidants. These antioxidants will be able to sequester the electrons and stabilize the hydrogen ions in those areas around the membrane. These are the antioxidants that the mitochondria will ensure you have enough of based off of being at a normal activity, healthy status individual. So let's take a look at this electron transport chain as it moves the hydrogens and electrons around the system. So we start with the development of the NADH, H+s, and the FADH2s. And so we get the NADH, H+s either coming from glycolysis or coming from the Krebs cycle. they interact with complex 1, stripping the hydrogens away, with the electrons following along. Notice the electrons are unable to pass through the membrane at a high enough velocity so as to keep up with the hydrogen ion. This is where CoQ will start to stabilize the molecules. We start interacting with complex 3. Notice the introduction of the FADs at this point in time. We start potentiation. We start pulling in secondary antioxidant molecules, which will help stabilize. In complex 4. Within each of the complexes, we have developed enough chemoelectrical charge so as to power up the ATP synthase and thereby generate the ATP and the water. So let's take a review of the ATP that we've been able to get from our pathways here. So we will get somewhere between 20 and 34 moles of ATP per mole of glucose that enters at glycolysis. A majority of that is going to come from the NADH pluses and the FADH2s interacting with the electron transport chain. From the NADH pluses, we're going to get somewhere between 25 and 30 total moles of ATP. From the FADH2s, we're going to get somewhere between 3 and 4 total moles of ATP. Which means that based off of pure efficiency, the electron transport chain is the most efficient. And glycolysis and Krebs will be least efficient. However, we have to remember that the pathways themselves are linked together in one continuous chain, where the ability to get all the way through from the initiation of glycolysis through the termination of electron transport chain is based off of the formation of the intermediates, the availability of oxygen to the mitochondria to allow for the continuation of Krebs cycle, and the electron transport chain.